Peace be upon you all, and welcome to another exciting episode with Dr. Fuz Raya about seeing God in biology. And we are on the molecular level talking about DNA. And today it's DNA data storage. So DNA is a data storage medium. But guess what? He's going to tell us that it's not only data storage medium for the genome, but actually some people are figuring out how to use it to store just data that we use. Is that the case, Fuz?
It is. This is, to me, a remarkable advance that's taking place in information theory and communication technology is to use the use of DNA as a data storage medium, which, people hope can actually help us address the data storage crisis that we're currently facing. But it also just highlights the remarkable design of biochemical systems. The fact that with DNA, its role inside the cell is to store information, and it is an information storage system in which the density of information is probably very close to the theoretical maximum.
Wow. And actually, it reminds me of a meeting I had with my staff who are like IT directors few years ago. I think it was like 2018. And I told them, guys, if you want to know the technologies of the future, you should check this. I talked about 4 or 5 topics, and one of them was actually DNA hard drives, or actually DNA data storage, because I was going through the literature, and somebody was holding a test tube, and the message was that this test tube full of DNA can essentially store everything that we have on the internet, a complete copy of the internet in this test tube. So this was like crazy. And there is this thing that people were talking about during this decade, that human knowledge used to double every I don't know how many centuries or whatever. And then in this modern age, the time for doubling of human knowledge is growing shorter and shorter and shorter. It's in the order of magnitude of months or something. And of course, okay, they're counting everything that humans are recording as knowledge. So if somebody takes a picture of his cat and stores it on his iPhone, it's essentially some more human knowledge. All the chats on, social media or WhatsApp, it's also human knowledge. But the matter of the fact is, all of this data needs to be stored somewhere. And we really have a problem with magnetic media because, magnetic media lose their magnetism after some time. And we need to throw things away, bring new equipment, fix errors, etc. So if this thing just keeps on growing and growing, the huge amount of information, high resolution cameras, high fidelity video, HD, and 4K, etc. So a short video of few minutes can be, nobody knows how many gigabytes in the future with all this crazy high resolution. And at some point of time, all the traditional kind of media that we're using today will be not feasible to be used or hugely expensive if we need to maintain fidelity of the data. And then I'm struck with this thing about using DNA as data storage. So I'm really excited that you would tell me today and the audience why DNA is so good in doing this. And can we really, at a point of time, think of a computer having some DNA material inside, or at least using it as mass storage or as cold storage, or the new tape drive within 20 or 30 years from now, or I'm not sure how many years. And this is very exciting. So I'm really looking forward to it.
Yeah, well, and as you're pointing out, it's not just simply the fact that the storage media that we have today will eventually fail, which means that you've got to make copies of the data that we currently have, but it's also the technology, it becomes obsolete as well. And so you have to then take data that is stored in old technology and update it to new technology. And of course, there's actually raw material that you need to build the actual storage
Mm.
media. And eventually, I've heard heard statistics to the effect that by 2040, we very well may be not having enough, high quality silicon, you know, on the planet to actually
Wow.
fulfill our data storage needs. And so,
Wow.
and it's interesting because some of the early thinkers along this line of using DNA as a data storage media note that, first of all, that life has been on Earth for maybe 3.8 billion years and has used DNA that entire duration of time to store data. And so
It's not getting obsolete anytime soon. Yeah.
it's not getting obsolete anytime soon. And and the thing is, we might develop new ways to read information in DNA or to write the information in DNA, but that the media itself is never going to become obsolete. So anytime there's a technology advance, it's going to be in terms of increasing the efficiency of reading or writing to the medium, but it's never going to be that the medium with DNA is obsolete. And also DNA is a remarkably stable molecule. The world's record, I think, is the recovery of ancient DNA fragments from a fossilized horse that was 500,000, 600,000 years
Wow.
old. And so, fragments of DNA, are able to survive in cold, dry conditions where the DNA is actually, complex to bone. That that can actually increase survivability. So people right now are actually looking at storing data in DNA and then encapsulating the DNA in glass and then storing it at cold, in cold temperatures under dry conditions. That may give you 500,000, maybe 1,000,000 years of integrity to the DNA system.
Wow.
Plus, you could
Looks,
also have
sounds like Jurassic Park to me. Huh?
Yeah, it does. It does. But compare
Yeah.
that to 10 years with a magnetic tape drive or tape system, right? And so anyway,
Absolutely.
and
Actually, it's a sad story to me because you and me will remember when there was, those floppy disk drives and
yeah,
the floppy disks, the 5.25 inch and
yeah,
the 3-inch ones,
yeah,
and as a student in high school, I had I was really the geek. Okay, so I had my 8086 and then an 8286, PC, and it had two floppy drives, a 5.25 inch and a 3 inch. And later, when I had all those nice programs that I've written, and the first hard disk that I bought was like 10 megabytes or something, and then those media went bad, and I lost what was very cherished and near to my heart programs that I have written, and I cannot get them anymore. So probably I should have asked somebody on the on the DNA storage to solve that problem 30 years ago.
yeah.
But okay, so let's see how this really works. What do you have for us today? Is that presentation or a discussion or what?
Yeah. Well, I thought what I could do is first of all start off with giving a molecular explanation for why DNA is such a robust data storage medium, because inside the cell, the role of DNA is to store data, and you want to have molecules that have a high degree of structural robustness and the capacity to preserve the structural integrity. And so let's go ahead and let me pull up my the slideshow, and we can go ahead and just talk a little bit about the structure of DNA and again
Yeah.
why it's such a robust molecule. And we've recorded a little short video just giving the viewers an opportunity to get some background information into the structure of DNA so people can refer to that. But I'm going to review some of the material
Yeah.
that's in that video just to help set the stage. And of course, this is a figure showing three different depictions of the DNA structure. And DNA is I like to say, is a molecule that has a ladder-like architecture where the uprights of the ladder correspond to the backbone of the DNA molecules that make up DNA, the DNA double helix. And then the rungs of the ladder are the side groups that extend from that backbone. And if, again, you twist it in a right-handed way, you get the well-known DNA double helix. And DNA consists of two molecular strands, and each of those strands is built from smaller molecules called nucleotides that are linked together in a head-to-tail manner. And there are four different nucleotides that are used to build DNA: adenine, guanine, cytosine, and thymine. And so here is a diagram of a typical nucleotide. This happens to be thymine, and it consists of three components: a sugar, a five-membered, a five-carbon sugar that forms a five-membered ring called deoxyribose, attached to what's known as the 5' position is a phosphate group, and then to the 1' position is the nucleobase, in this case thymine. And it turns out that the way the strands are built is through essentially what's called a phosphodiester linkage, where the phosphate group attached to the 5' position of one nucleotide will react with the 3' OH group of another nucleotide to form this phosphodiester linkage, and that's repeated over and over again to build each of the individual strands of DNA. And the point that I want to make here first is that the structure of DNA is highly optimized specifically for information storage. So this makes a lot of sense. If DNA is going to be stored, is going to function as an information storage system, then, of course, you would expect, or you would hope, that the structure is optimized. But it turns out every detail of the DNA molecule structure contributes to essentially its stability and its role as an information storage system. So in many respects, it's no wonder that technologists are turning to DNA, again, as an information storage system. So, for example, the phosphate...
I don't want to jump this, but I'll just ask you this question, then maybe we'll refer to it at the end. When you say it's optimized, do we have evidence that it is optimized to the maximum of the maxima, or just to a good level of optimization?
Yeah, that's a great question, and in many respects, I don't think we can answer that question. We do know that it is a highly optimized system, and we can compare DNA with alternatives, with conceivable alternatives, and show that none of those alternatives work as well as DNA. And in fact, I'll show you some of those potential alternatives as we in the next few minutes. But I don't know that we can necessarily say quantitatively that the degree or the level of optimization. So it's really a qualitative statement that I'm making when I make that statement. Now, maybe there's somebody that's figured out how to quantify the optimality of DNA, but I don't know how to do that. But I wouldn't be surprised if there's somebody that does.
in terms of data storage, would there be a theoretical ceiling of how much information can be stored on a given amount of material that we can then say, well, DNA seems so close to that?
Yeah, I think that is the case, and I've heard people make the claim that DNA is very close to the theoretical storage limit. And you can get a sense for this, as you were mentioning earlier. A test tube of DNA, can store all the data that's on the internet. Another statistic I heard is that a kilogram of DNA can store all the digital data that exist in the world today.
Wow.
And
Wow.
so, that gives you, a sense for the information density that can be built into DNA. It's you're never going to, if you have a dump truck full of DNA
Ah,
powder, right? I
ah,
mean,
that
it's
would be
hard to
crazy.
imagine, how
Yeah.
much, how much information you could actually store in that. It's probably beyond what we could reasonably create, so
So what we at least know is that it is orders upon orders of magnitude better than the best thing that we did in terms of our own designs.
that's that.
This we're sure of.
Yes, that's exactly right. Yeah.
All
So
right.
then, let's take a look. For example, one of the questions is, why is phosphate in the backbone? And the reason why this is a pertinent question is because there are other possibilities that could be used. For example, sulfate could form a sulfate diester as opposed to a phosphodiester. You could have a sulfol diester, or you could use arsenate, which is a direct analog to phosphate. On the periodic table, arsenic is below phosphorus, and its chemistry is going to be very similar. So these would be two uh, reasonable possibilities to be used in place of phosphate in the DNA backbone. Well, it turns out that phosphate, not only can it form two bonds, but it retains a negative charge. And that negative charge actually stabilizes the DNA double helix, because when you look at the nucleotide, the sugar and the phosphate are water soluble, and the nucleobase is hydrophobic or water insoluble. And so the interior of the DNA is hydrophobic. The backbone of the DNA is hydrophilic. So it kind of is like a detergent, but that means in an aqueous environment, you actually have essentially the interior of the DNA free of water, which is actually critical for the hydrogen bonding interactions that take place between the nucleobases. But that you get structural stability from the negative charge of the phosphate because of that um, amphiphilic character to DNA solubility properties. Plus, it also serves as a place where proteins can bind, which allows the cell to actually access that information in DNA. Now,
But
sulfate can go
yeah,
ahead.
I was going to ask, if what about sulfates and arsenics? Yeah.
Yeah, well,
Hmm.
yeah. So sulfate can form two bonds, but it doesn't bear a negative charge, and so you don't get the stability of the double helix, let alone
Hmm.
its the chemical properties that again allow for protein binding. Now,
Wow.
arsenate won't form stable bonds with organic materials. So organic arsenates are not very stable compounds. So phosphate forms these stable organic esters and has a negative charge. And I don't know that there's anything else that could be used in the place of phosphate that would have, again, those kind of properties. So you can
Wow.
begin to see that the structure of DNA isn't arbitrary, but there's a rationale with respect to the structure of the backbone. Well, why deoxyribose? And
I really like those why questions,
yes, yeah,
because
me too.
when somebody asks why and always finds a rational answer, it's telling. It's telling that from all the options, like in the previous episode, why ATCG, there is an answer. Why phosphate, there is an answer. Why deoxyribose, I'm sure there will be an answer. And it tells that there is a designer behind that, because everything has a why.
Yes, exactly. And it turns out that um, it, in addition to DNA, there's a sister molecule that's used in the cell called RNA that has many of the structural similarities to DNA, but some significant differences. One of them is that ribose, they both are five carbon sugars that form five membered rings, but in the two prime position, ribose has an OH group, whereas DNA doesn't. That's why it's called deoxyribose. It means that it's missing an oxygen. And it turns out that that OH group is missing in the 2' position. Well, this turns out to be significant because having that OH group in that prime position actually causes RNA to be unstable, because that OH group can actually attack the phosphodiester linkage, causing a self-hydrolysis. And so because DNA doesn't have the OH in that position, it's inherently more stable, where it's not going to undergo that self-hydrolytic reaction. But it also turns out that people have done these studies that a 3' 5' linkage is more stable than a 2' 5' linkage. We mentioned the self-hydrolysis of the DNA chain. Also, removing that OH group creates space in the interior of the DNA molecule that allows the interior to accommodate the nucleobases. If you have an OH group, it's very hard to form a stable duplex because that OH group is, again, creating what are called steric problems. Also, that removing that OH groups creates structural flexibility in the deoxyribose, which actually is very important because it permits proteins to bind to the DNA. That flexibility in the DNA allows kind of the active side of the protein to be able to fit with the DNA, kind of a hand in glove mechanism. It allows the DNA to be methylated, and then it allows the DNA double helix to remain intact when DNA damage takes place. And the reason for all three of these things has to do with something called Hoogsteen base pairing. It turns out that the base pairing that we see in DNA is called Watson-Crick base pairing. But if you take one of the nucleobases and you rotate it 180 degrees, you can set up a different hydrogen bonding pattern. This is called Hoogsteen base pairing, and it's extremely rare, but it does occur in DNA, and it tends to occur in DNA in regions where there's methylation or protein binding or DNA damage. And that actually, that Hoogsteen base pairing under those conditions helps to stabilize the double helix. And you know, the chemical reasons for this become very complex, but the point is that having that missing OH group allows for this really unusual base pairing that plays a very important role in retaining stability of DNA.
So you mean that even when a problem happens and there is damage, the possibility of the other type of pairing keeps the DNA together until the damage is fixed?
Yes. And so, for example, if you have an RNA double helix, which you can form, because of that OH group, the RNA cannot engage in Hoogsteen base pairing, the RNA duplex. It only can engage in Watson and Crick, but it means it's very difficult that RNA duplex won't tolerate damage, right? The damage becomes far more catastrophic. So it's one thing for the damage to happen. You still want the helix to retain integrity. You want it to be distorted enough that the cell's machinery can recognize the damage, but you don't want the helix, the integrity of the double helix to be compromised, right?
Wow.
While you're waiting the repair, right?
Yeah.
So
I think this on its own, this is just a side note, is pretty good arguments against the RNA world hypothesis, because I cannot imagine a cell that is based on RNA genetic material surviving if RNA can self-hydrolyze, stick to each other in many places, distort itself without return, and then this cell will just keep on living.
Yes.
Especially there isn't error, the kind of error correction and detection mechanisms that are available to DNA machinery versus RNA, so it will just be irreversibly destroyed. And yet some would still argue that life started with RNA and without any evidence.
Yeah, excellent points. Yeah. Why adenine, guanine, cytosine, and thymine? In the last episode, we talked a little bit about why adenine, guanine, cytosine, and thymine. And it turns out that these 4 nucleobases actually display very unusual UV stability. In other words, DNA is constantly being exposed to UV radiation, and these 4 nucleobases have chemical properties that allow the bases to absorb UV radiation and then radiate it away without actually damaging the base itself.
Wow.
They're much more efficient at radiating away that excess energy without causing chemical bond breakage. And then on top of that,
I'm thinking that if DNA used something else, then plants that are seeking the sun for photosynthesis would have had plenty of trouble.
Oh
That
yes,
would have terminated life on Earth in its cradle,
Exactly. That's exactly right. And then of course we talked about this point is that because these are the 4 nucleobases that actually form as even bit parity codes, and that actually imparts an error detection capacity to DNA that wouldn't actually exist otherwise. And then, another question is why thymine in DNA and not uracil? Uracil is actually used in RNA in place of thymine, and uracil is a chemically simpler molecule than thymine is. And so this is showing the difference between uracil and on the right, and then thymine in the middle. And you can see there's a methyl group. But it turns out that the reason why thymine is used instead of uracil is because cytosine undergoes what's called a deamination reaction spontaneously, where thymine will actually undergo a degradation through deamination to form uracil. And when that happens in DNA, if that happened in DNA, then you would have a uracil where a cytosine should have been present. But it turns out, and so when that happens, because thymine is used instead of uracil in DNA, that uracil that shows up in the DNA double helix is recognized as being a mistake and then can be removed, can be replaced. That error can be corrected. Also, if thymines are adjacent to each other, they can actually absorb UV radiation, forming thymine dimers, and then that is something that can be repaired or corrected for. So this is another way in which you get kind of an enhanced UV stability. And then there's also a reason why the strands of DNA are anti-parallel. We talked about this in our introductory video where we
But for the uracil in the RNA, is it the case because RNA is intended to be short-lived? It's like transcribed, and then some proteins will be produced, and then if it degrades, it degrades. Then another DNA will be transcribed, and another gene will be transcribed.
That's exactly right. So, by using uracil in RNA, that's a really great point. Even though the difference between uracil and thymine involves a single methyl group, when you think about the number of uracil molecules that are used on an ongoing basis to make RNA, that would become energetically expensive if thymine was used instead of uracil. And as you're pointing out, the RNA is going to be degraded anyway. So, why not use the cheaper, less expensive option?
The cheaper version. Yeah. And I'm also thinking that, RNA is like, I like to give it the parable of a program in memory. All right. So you load the program from the DNA, you load it into memory, it does a function. And then, after it does the function, it can just go away. And I'm thinking if RNA was really stable and it will just keep piling up, and the cell will need to destroy it, to exert further energy to destroy it, rather than it, degrading itself, making half of the job or something, and then the degradation mechanism will take care of the rest. So it's even catching one's attention that the right components in terms of half-life or auto degradation are used for RNA that needs to go away, but the more stable ones are used for DNA. So everything is fit for its use or its purpose.
Yes, and this is why the while it's not desirable to have the OH group in the 2' position in deoxyribose because of the self-hydrolysis of the DNA backbone, in RNA, it's actually preferred. It's actually a positive design feature because, again, you don't want the RNA to persist in the cell. You want it to be to have a very short, half-life, as you said, or very, you want it to be an unstable molecule or have minimal stability.
Yeah, amazing. All right.
And then
anti-parallel.
Yeah, and we talked a little bit about this, is that there's an orientation to the 2 strands of DNA, kind of a top to bottom orientation. It's biochemists call it 5', 3', 3', 5', and the strands are actually anti-parallel, having opposite orientations, and so this is a diagram showing the different directionality of the 2 strands, and it turns out that this again can be shown to be really important because the anti-parallel arrangement actually forms a more stable double helix, and it also creates topological effects in DNA that help and facilitate protein binding. So the bottom line is, when we look at DNA, it is a highly stable molecule where, the point is that every detail of the structure of DNA actually contributes to its stability. It's not accidental. It's not, a historically contingent outworking of random processes that just kind of settled on DNA as the information storage system. But there is a deep-seated rationale to every aspect of the structure of DNA, where even the minutia of the molecule, is playing a critical role for its stability. So in many respects, that what you see here is that insight from this asking the why questions really reinforces, the decision of technologists to choose, DNA as a data storage medium. You're not going to be able to probably design a molecule that's better than DNA for data storage. Now, you might design molecules that might be more useful for DNA, because one of the limitations of DNA storage is it's very time consuming to write and expensive to write it to DNA, because you've got to literally synthesize DNA, and it's also time consuming to read out that information. Now, people are working on coming up with clever approaches to writing and reading DNA more rapidly. So right now, DNA as a data storage medium is ideal for long-term storage, for archival storage. It's not good for, where you need to access on an ongoing basis that information. But some people are looking at using DNA as an inspiration to design molecules that may not be as effective as DNA at storing data, but might be more amenable to, easier and quicker, more efficient reading and writing. And I don't know if people are going to be successful in developing these systems, but there are people exploring those possibilities. But nevertheless, DNA still is serving as that template, that inspiration for, where the principles of how DNA works and how it stores information are being utilized in these alternative systems.
And probably the difficulties that we are having is because we don't have the same technology that the cell has, because the cell is doing it all the time, reading, writing, replicating, transcribing it all the time. And we also see that in the cell, the volatile pieces of information are actually encoded on RNA, not on DNA. So probably because RNA is, as you have taught us, is pretty unstable, So if we try to use it, it will be even more difficult to us because of its instability and self degradation. So probably if we are to design an organic computer, we will essentially end up doing what the living organisms do. We will use DNA for hard disks, and we will use RNA for processing in memory, sort of, or
Yeah.
a mix of RNAs and proteins or something. For proteins would be like rendering the results or something like screens. So it turns out that what the body is doing or what the organism is doing is essentially the ultimate design,
Yes,
sort of.
exactly.
Yeah,
Yeah, because the entire biochemistry, to your point, the entire biochemical machinery devoted to reading and writing information in DNA is it's a very complex set of machinery to begin with that are
yeah,
highly sophisticated, but it's designed specifically to read and write that information in the context of the cell. And it's incredibly efficient and incredibly rapid. It's just that what we're trying to do is interface our technology with the cell's technology. And it's that interface where the inefficiency is existing right now. Right. And I wouldn't be surprised if the way around that inefficiency is to copy what the cell is doing, is to making use more and more of the cell's machinery.
I would expect so, because DNA is this way because it has a certain purpose. So if we want a different purpose, then we might as well go back to biomimicry and see how the cell is using RNA, and maybe 100 years from now we can mimic it or something. I don't
Right.
know.
Right.
Yeah, so it's amazing, and I want to get your insight about this. I know we're gonna make soon enough an episode about the genetic central dogma and the transcription, translation, etc. But I'm just thinking this. So we have a living organism. The living organism needs to live and pass on its traits and manifest certain phenotypes that need to be stored somewhere. And then there's this crazy system that reads the data, what you have explained, like using phosphates that where proteins can bind, and phosphates have this negative charge that can make it like water, and all the other stuff. But we have a system that needs information, and then it has all the machinery to read the information, and it has the medium that is amazingly suitable to store the information, and to bind to the machinery that reads the information. Once again, it's like the sender-receiver issue, where there has to be this full conspiracy, otherwise nothing will work. A very stable molecule is storing information. An unstable molecule is like taking a copy to execute it temporarily and then go away. Machines that are custom built, helical structure that protects hydrophobic interiors, etc. In your mind, as a biochemist who really sees the details of this, and also a question about science, is there any explanation today about how DNA came about?
Not really. I mean, what you see, in terms of chemical evolutionary models are attempts to maybe explain how the different components of DNA might have evolved, through chemical evolution, and, speculation on how maybe those might have combined. But there really is not, a robust explanation for, how you would wind up with DNA. It's one thing to have, ribose form through something called the Formose reaction, but deoxyribose doesn't form through prebiotic routes, to my knowledge, and
Uh,
so
yeah,
so even getting some of the components is problematic. So there's
yeah,
not a robust explanation for how it comes about, let alone how you get information, encoded into the first DNA molecule.
But even put, so let me tell you this. Let's say that somebody figured that the magnetic material of a hard disk can come about by chance in the middle of a desert, or in the middle of a forest, or somewhere in the ocean. But if I find that if somebody tells me, but guess what, there is also a way that a piece of magnetic material comes together in the form of a disk that is made of tracks, and then also a reading arm that uses, I don't know, laser or something to read it, will also come about. Now I'm thinking he's crazy. But now if he tells me that all those components will come about together in the same place, get into a case, and a data bus will come to that, now I'll be calling the ambulance. Because it's completely crazy, because there is a complete conspiracy.
Yeah.
Not only that DNA exists, set aside how to reach its components from prebiotic, I don't know, chemistry, but also that those components come together, that the phosphate and the sugar and the nucleotides, and then it just forms the strand. And then what happens if the strand is formed and there is nothing to read it?
Yes.
And where's the container and everything? So this is and another question is, if this information gets into the DNA, there's also all the other details about where the information starts and ends, right? It's not just like a series of bits like sitting out there. There's like a directory structure, a system to show where the files are and everything. Yeah, so completely amazing. So what else do we have on top of? Did somebody really try to store information on DNA today?
Yes. Well, and by the way, and I think we've mentioned this already, but the information density, that can be stored in DNA is astounding. 1 gram of DNA can store 455 exabytes of data. And as I mentioned, 1 kilogram of DNA can store all the world's digital data. So the information storage capacity is through the roof. Now, in the cell, and we'll have a whole episode devoted to what's shown here, which is the genetic code, where the information in DNA is defined by a set of rules that relates the sequence of nucleotides in DNA to amino acid sequences that are found in proteins, and a coding triplet is used, where combinations of 3 nucleotides specify different amino acids, and so there's 4 to the third, or 64 coding units. But there's no reason why you can't define your own code in order to store information in DNA. And so this is a diagram kind of showing conceptually how that data storage works, where you have digital files, and they can be text files, or photographs, or audio or video files that are ultimately a sequence of bits, ones and zeros, and you can then use different nucleotide combinations to essentially define digital words. So you're developing your own coding system to encode that information. And then once you do that, you just simply synthesize the DNA. And then if you want to read that information, you'd have to just sequence the DNA. And then you could then convert that the sequence data back into digital data, and to and recover those files. So it's a very simple concept, but in effect, this is exactly the concepts that the cell is using, as we'll see in a few weeks when we start talking about the central dogma. This is exactly the same concept that is used in the central dogma of molecular biology. The one of the most important proof of principle experiments towards this end was done by George Whiteside's group at Harvard in 2012, where they encoded a book consisting of 5,300 words and 11 JPEG images into DNA molecules. And what they did is they actually produced fragments of DNA. So it wasn't just a single long DNA molecule, but they had fragments where each of those fragments had index sequences that would function like the page number in a book.
Wow.
So that you could actually fish out individual pages from the book, as opposed to having to sequence everything. You could selectively sequence those fragments, fish them out and sequence them. But they also made redundant copies, so that way if there was damage taking place in any of the individual fragments, you would have redundant copies that would allow you to recover that information. Because again, DNA is a chemical material. It will undergo degradation to some degree, but the hope is that by having multiple copies of each of that information, and because DNA has such a high information storage density, you can have thousands of copies of that data file, and it would take up probably no more room than if you had a single copy. So why not do that? Now you've extended the lifetime of DNA as a data storage medium through having redundant copies, again, storing it in dry, oxygen-free, cold conditions. And then again, there's people that are encapsulating DNA in glass beads, where the glass itself helps to preserve the integrity of the DNA molecule, which is essentially the same mechanism that preserves ancient DNA in fossil remains. It's the minerals that help to preserve the integrity of DNA over vast periods of time. So it's it's again it's a remarkable system, in terms of its data storage capacity. And to me, this work really is theologically significant, because I've heard a lot of skeptics say, well, look, really and truly, there's not information in DNA, right? We're just using these metaphors, to help us understand what's happening, in this physical chemical system. And you're just taking that metaphor to an extreme by arguing that there's real information there, or that information is, structured in the way that we would structure information or manipulate information, that this is just, a metaphor without real foundation. But when you see this idea of using DNA as a data storage medium being part of technology now, and the principles that are being used are identical to the principles that are used in the central dogma, it's very hard to argue that this isn't real information.
It's not metaphorical anymore. It's not metaphorical. We're using it.
Yes. Yeah. We're using DNA in a different context, but yet in the same conceptual scheme as inside the cell. And it's only possible because it really is. If DNA is a data storage medium in technology, it's a data storage medium in biology. You can't have this fractured way of thinking and be reasonable.
Yeah, absolutely. You reminded me of something. I was having a discussion with an atheist at the point. I go into those discussions a lot. And he said, well, if maybe somebody would analyze DNA and find in some of the sequences, God's name written there, or a signature or something. And I'm thinking while you're talking now, if this is not God's signature, what else is?
Yeah, right.
What do you want exactly? If this is not the signature of a designer, then what is? Yeah. And if this is something that somebody would find as a relic of a previous civilization or something, that they're using DNA storage, let's say, we discover that, what have you, pharaohs or ancient Chinese or something, where we're having this secret technology and they're using DNA storage. Nobody will say this is just a coincidence, and this machine is there because they were just playing with things and they just created a DNA storage machine. It's really ridiculous when I think it's really ridiculous when people see this and somebody will still say this is a metaphor and you're taking it too far. It's unfathomable that somebody will think this way. It is really bio inspiration, and it's amazing because I also was thinking when you were talking, and I was thinking about my floppy disk drives, and I'm thinking that I am way more than 10 years old, and when a scratch happens in my skin, still I get my same skin. If I burn my fingers, still I'll get my same fingerprints, and the same proteins. I'm not turning into anything else, and it means that this system is like 4 decades going into fifth, okay, and and still maintaining itself way better than the best things that we have, and it's for sure a great foundation for biomimicry and bioinspiration for sure. That was really an inspiring and very intricate presentation, Fuz.
Thank you, thank you. You know, there's, as a biochemist, I've been a biochemist for over 30 years, and I still am discovering things about DNA and learning things about DNA that boggle my mind. There again, it's so impressive to me that every little detail of the DNA molecule makes exquisite sense. It's just not accidental. It's just not arbitrary. It's just not the way it is. There's a reason for it, and the more that we're learning, the more and more elegant the rationale is that's being exposed with respect to the way biochemical systems operate.
I want to ask you this. Did you see people who when they've studied this, it brought them to the realization that God exists and he cares?
Yes, in fact, I was one of those people.
Okay. Okay.
I was an agnostic. I was an agnostic when I started graduate school, and it was, the elegance of biochemical systems that convinced me there had to be a mind behind everything.
Wow.
And what was interesting to me is that as a graduate student, as I would interact with other graduate students and even interact with my professors, it was not uncommon for all of us to marvel at the way biochemical systems work. You would hear people say, "Look how cool this is! Look how amazing this is!" And it's those are code words for for design.
Yeah.
Right? That there's a divine intuition that you're reacting to.
Absolutely.
Or you have Francis Crick, who died as an avowed atheist. In his autobiographical account of discovering the structure of DNA, called "What Mad Pursuit," he admonishes biologists by saying, we constantly have to keep in mind that what we see was not designed, but rather evolved, right? And so his admission there, the tacit admission, is that these systems really do look like they're designed, and you have to almost in a sense insist on a materialistic interpretation, to explain away that design. But the use of design language permeates the biochemical literature. You can't avoid it.
You can't avoid it.
Why some people see the obvious evidence for design for what it is and why others don't. That to me is a mystery. But the hope is to get people to stop and think for just a minute, about.
But is it really that they don't see it? Because this statement of Crick is really telling, because he says yeah, it looks so designed, but we need to remind ourselves that it's not. So it looks so designed. So you see the evidence that it's designed. What evidence do you have that it's not? And it's like you stand in front of the mirror every day and say, biochemical systems are not designed like 100 times or what? Positive reinforcement or what? This is psychological tricks. This is not how science should work, right?
Yeah.
So is it that they're not seeing it, in your opinion, or they're intentionally denying it?
Well, and again, I don't think anybody disputes the fact that these systems are remarkable systems, that they have the appearance of design. The only other option is to somehow argue that evolutionary mechanisms are responsible for that design, as opposed to, a creator, right? This is the blind watchmaker claim, that Richard Dawkins popularized, that evolution is the blind watchmaker. But I when it comes to biochemical systems, it ultimately is the origin of life that is the way you explain the genesis of biochemical systems. And that's where even people that work in that field are, readily acknowledge we have no understanding whatsoever how life could have evolved through chemical evolution. There are
Yeah,
ideas that people are proposing, but they're not very compelling ideas, and they don't withstand, experimental, analysis, experimental, evaluation whatsoever. So, to me, I think the data forces you to think that there's some kind of genuine design if you really are honest with the data.
that's been really an inspiring thing, and I hope this whole series and these episodes bring really people who are experts to stand in front of the mirror and tell themselves in the eye, it is really designed, and we know it's designed, and we're going to change our stance. And people who are not really experts, to know that even the experts realize that it is designed, but they just choose not to accept it.
Yeah.
And to see that what is said in the media or by science explainers is something, and what is really the reality and what the science points to is completely different. And that saying that this might be chemical evolution or evolution in action is really a philosophical stance rather
Yeah.
than a scientific stance. Would you agree to that statement?
I sure would. I sure would agree with that statement. And uh, there's something that you said. I think the last episode that is really, I think, profound. That I'm going to use. So I'm going to appropriate it, but I will give you
You have
credit.
you
I'll
have
be
a
happy to
license
give you
to.
credit, uh, but it's the fact that when we ask the why question, and we get an answer, that in and of itself is absolutely profound. Because if everything is ultimately the product of unguided, historically contingent, mechanisms, then we can ask the why question, but the answer should that we get should be there's no real reason, right? But if we ask the why question and we suddenly see that there's a rationale, and it consistently we see that there's a rationale, and no matter how deep we go or how broad we go, we continue to see a rationale. That to me is suggestive that there is ultimately a, a mind behind everything, because it's the why is leading us to the purpose, right? And
Absolutely.
purpose comes from a mind. So I really very much appreciate that point you made. It's impacted me. It's sticking with me, and I think it's real insightful.
Yeah, thank God for that. And plenty of what you have said is sticking with me already, and so many great insights. First, this has been a great episode, and I hope so many people will make benefit of it. So may God bless you and give all of us the light to see the truth and to see the great wisdom and glory of God, our Creator. And I'm really looking forward to the coming episode. And those who have written questions on the live stream, please do jump onto the after show, where we'll have a fun discussion about what we have learned today. Thanks a lot, Fuz, for your teaching, and I'm really looking forward to next Thursday.
Thank you. Same here.
Thanks to everybody, and peace be upon you all, and see you again soon. And don't forget to send us questions for the next episode, God willing, and peace be upon you all.